You are listening Audio Paper Series with Yasir, and today's guest is Dr. Zamudio from Quadram Institute, Norwich, UK, to discuss one of our publications, Dynamics of Extended Spectrum Cephalosporin Resistance Genes in E. coli from Europe and North America. It is published in Nature Communication. Welcome, Roxana.
Hello, Yasir. Thanks for having me here.
Good to have you here. What are extended spectrum cephalosporin resistance genes, and what is the purpose of this study?
Okay, thanks for the question. So the extended spectrum cephalosporin resistant genes are sometimes found in the bacteria and are responsible for the resistance to the third and fourth generation cephalosporin antimicrobials. So this means that the bacteria do not respond to these antimicrobials. So also, I should mention that these extended spectrum cephalosporin resistant genes encoded beta-lactamases are responsible to inactivate the antimicrobials. And there are two types of resistant genes, which is the ESBL and the MPC. So the purpose of this study is to determine the dynamics and the evolutions of these resistant types, the ESBL and the MPC, over time in different ecological compartments, such as country and host species, and to identify potential factors contributing to the spread and persistence of this extended spectrum cephalosporin resistance.
Okay, so basically the purpose of this study is to find that how these extended spectrum beta-lactamases are spreading across different countries.
Yes, different countries, also different host species.
Okay. So how did you carry out this study?
Okay, so in this study, we have the opportunity to work with key collaborators from Canada, France, Germany, and the UK. So this network of scientists brought collective expertise in genomics, microbiology, epidemiology, and human and veterinary medicine. So through these international collaborations, we gathered a whole genome sequence from large collections of bacteria that was collected for many years. So our strategy was to select bacteria randomly from these large collections from our partners.
Okay.
And then also we selected these based on the phenotypic resistance and not on specific genotype. So we have representative informations of these genes in the general populations during that time frame that we are investigating. We examined the whole genome sequence, around 2,000 resistant bacteria. Predominantly, there are Escherichia coli from North America and Europe. And as previously I mentioned, so we not only look at to the bacteria from different geographical regions, but also we look at to different host species, such as humans, animals, food, and wastewater.
Okay. All right, so the same organism acquired from different sources, so kind of one health situation.
Exactly. We use this one health context to understand the relationship between these ecological compartments to understand the spread and the persistence of the resistance.
Okay, nice. So would you brief us with the one or couple of main findings of this study?
Yes, so we have two main findings. So the first main findings is that we found the dynamics of the ESBL and the MPC, which are the type of genes, they changed over time in our study period. So, for example, in the 4 animals from Canada, the MPC were predominantly at the beginning of the study, while the MPC was predominantly in humans and 4 animals in Europe. But way through the study period, the ESBL emerged and expanded in North America, and the MPC was detected and increased in the humans from Europe. So basically, we confirm that there is a contrasting trends on the dynamics of the ESBL and MPC in North America and Europe.
Okay.
So our second finding is that we found differences on the relative importance of the plasmids versus clonal expansions for the spread of this IMR. So, for example, we identify specific successful bacterial lineages and plasmid types and subtypes, and they are major drivers of the disseminations of this resistance in Escherichia coli worldwide. So with that, we learn that there are plasmid subtypes that could cross borders between different lineages, host species, countries, and bacterial species.
So what are the applications of these findings in science and community medicine?
Yes, I think this is very important. So I would like to mention that examining the antimicrobial resistance in the global One Health context is very important to gain knowledge necessary to combat the spread of the IMR. So our strategy that we use in our study can be applied in other pathogens as well as commensal bacteria to track the IMR between countries and host species. So this knowledge will contribute to design target surveillance studies, for example, specific plasmids or lineages. And also it will contribute to the interventions to reduce the spread of the antimicrobial resistance.
Okay, so you mean targeted surveillances are important for the containment of the spread of antibiotic resistance. Very good concept.
Yes, yeah, exactly.
Lovely. Thank you very much, Roxana. It was lovely talking to you, and thank you very much for sharing very interesting piece of science.
Thank you very much for this opportunity, Yasir.
Cheers.